There's been a lot of talks about AI, and this is going to be an attempt to try to bring some natural intelligence back into the mix here. Really, the big goal is we've all heard of brain-machine interfaces, but to move beyond brain-machine interfaces to what we would call brain coprocessors, machines that not only can read out from the brain and put information into the brain, but ideally do something interesting, computational, reactive, whatever it takes to treat a disorder or to improve mental function. And so, really, what we're talking about here is a two-part problem. One is to develop a suite of technologies that allow us to record and deliver information from the brain as precisely as possible. And that means both invasive and non-invasive methods. The second part, though, and this is part of why I was eager to come to this conference to talk with a lot of really interesting people on this side as well, is how do we start to develop the algorithms for detecting a state, for example, generating a hypothesis, and then intelligently supplementing what the brain is doing, both to repair pathology and eventually to augment human cognition. So this is sort of a schema of this idea here, where we collect data, we detect what needs to be detected, we react, and we perturb. The problem space is a big one, and many people, I think, especially neurologists and psychiatrists, are of the opinion that a lot of these disorders represent large unmet medical needs. Stroke, addiction, chronic pain. Many of these disorders, such as Alzheimer's and stroke, are what we might call barely treatable. Others, like blindness and deafness, people are struggling to think about how to develop technologies to confront the complexity of the loss of a sense or an ability to move in the case of a stroke. But in this audience, of course, we also want to think about how can we augment functions as well. And in many cases, they're the same thing. For example, if we were trying to come up with a way to deal with anxiety, and at the end I'll show you some data from our group on this topic, then there's also the ability to prevent anxiety, to augment, to give resilience, to prevent stress from having a deleterious effect on cognitive emotional coupling. Now, one of the big problems, of course, is that the brain is extremely hard to engineer. If we think about an abstraction layer for the brain, much as if we were trying to engineer a piece of software or to build an appropriate computer system, what we find is that it's very hard even to know where to begin. For centuries, we've known about brain regions, Broca's area, involved with the generation of speech, for example. And over the last 100 years or so, scientists have been zooming in on the brain, looking at the cells, the individual circuit elements of the brain, and their subparts too, their inputs, the dendrites, and the output, the axon. And within, a highly sophisticated electrical computational machine capable of taking thousands of inputs and distributing them to thousands of outputs. Now, a big area of research over the last 2 decades or so is looking at the types of cells in the brain. And unlike other organ systems, like let's say the heart, which has really one dominant cell type, the cardiac myocyte, the brain contains thousands, perhaps even more types of cells. And in fact, a large and growing discipline of connectomics is starting to look at these cells and trying to look at the electromicrographic structure of these things and finding that there's no room in the brain. It's all packed in there. A cubic millimeter has millions of cells and billions of connections and so on and so forth. Now, one of the advantages of thinking at the cell level is that it's a nice intermediate middle ground, so that's this middle column here. But also, pathologists have found that many cell types are indicative of different disorders. So for example, these chandelier cells here are one of the cells that are most atrophied in the schizophrenic brain. And other interneuron types are lost in epileptic tissue. So what if we could try and figure out ways to engineer these cells, which still, while an acceptable first abstraction layer is still very complicated, right? We have 100 billion of these computational elements in the brain, and they are continuously computing at the millisecond timescale. So other branches of synthetic biology, such as gene regulation, are dealing with problems that are about 1 million times slower. How are we going to deal with the ability to control and read out from, let's say, the entire brain over some reasonable timescale? This also highlights, I think, one of the key success stories of the 20th century and why it might meet with roadblocks in the 21st, namely that of pharmacology, antidepressants, anti-epileptic drugs. These drugs bathe the entire brain in a substance, for the most part, and that will affect both the circuits that need to be addressed for therapy and also the circuits that don't, and that can lead to side effects. So neuromodulation, the use of energy to perturb neuroactivity, is a very tantalizing and promising area. So here's my one slide trying to explain all of neuromodulation theory in one PowerPoint here. So here are 3 neurons that are connected. You can kind of see activity propagating about. The resting state is color-coded purple. Neurons are normally negatively charged with respect to the world, and you depolarize the neurons or bring the voltage up when they are active. And so what's happening is when one neuron, let's see if we can use the mouse here a bit here, when one neuron sends information across this gap, you have chemicals being transmitted, and that it causes the depolarization of the next neuron. So if we were able to zoom in on this, you would see something like this. You have a channel in a membrane, and there's positively charged ions outside. And when a transmitter, and the voltage is flat, right? It's not much happening in this case. And when a neurotransmitter arrives, you can actually get those channels to open. The charge flows in, and you get a spike. And then, of course, the transmitter will eventually move away, and it'll go back to the resting state. So one of the big questions we began with was, can we start to program in patterns of activity through very precise cell-specific activation? And if you could program in activity cell by cell, you could start to test the efficiency of that pattern to generate a computation or a behavior, or even perhaps someday a subjective experience. But also you could start to reprogram corrupted neural computations. If one cell isn't carrying its weight, we could try to put that information into the downstream cells. So one of the ideas that we have been working very heavily on is, can we use light as a very precise sculptable method for sculpting neural activity? In other words, can we simulate this effect here where we fire a spike with a chemical with a pulse of light? So the ability to use microscopy in neuroscience is a very powerful one. These are 2 photon images, deep images in the brain, and these kinds of microscopes have been used both in basic science, but also a lot of people, especially endoscopists, are getting interested in using these clinically. And the goal is, what if we start to use light to turn these individual cells on and off? On the right hand, I just color-coded some of these cells red, and on the left, what if we could beam light to those specific cells? So really the modern story of this begins with the discovery in 2003 by Georg Nagel and Ernst Bomberg and colleagues of a blue light-gated ion channel. And what that means is it's a molecule that's found in a green alga, which it uses to drive its flagella and steer around, which binds a ubiquitous vitamin A derivative, so it can work in normal tissue. But when you light it up with blue light, it lets in charge, just the same way that I showed you on that previous slide with the neural network firing. And more importantly, perhaps, we can genetically target specific cell types, just the cell types that are involved with epilepsy or Parkinson's or whatnot. And we can do that by taking the gene for this genetically encoded protein, putting it into the right vector, making a virus, and popping it back into the cell. And when we do that, we can see it lighting up on the cells. Here you can see a nice cell here with it on the border. And when you beam light at it, you can get them to fire spike trains, very precise ones. So here are 3 spike trains, not unlike the ones in your auditory cortex right now when you're listening to me, that are firing in response to pulses of blue light, which are those little dots underneath the spike trains. Now, in basic science, these are being used very widely in different animals to test the sufficiency of a given pattern of activity in generating a behavior. So you can put in, for example, a pattern into the mechanosensory neurons of an animal, and it will respond as if it had been touched, and so on and so forth. But I wanted to highlight a new clinical area that I thought would be of interest to this audience. And this is an example of a form of blindness where people have lost their photoreceptors. So you can see here, in the normal retina, photoreceptors, and then in people with retinitis pigmentosa, for example, the lack of those. Now, several groups, including ours, have been pursuing the use of these opsins to treat these kinds of disorders. This is all collaboration with the labs of Alan Horsager and Alapakam Sampath at USC and Bill Houseworth at Florida. And here's an example of a mouse. It turns out that the albino mice you see at pet stores are naturally blind, and the blindness mimics that of the human, where we've delivered the gene for these to the retina and been able to sensitize it to light. So this is of interest because there are lots of ideas in the neuroprosthetics field to take a camera and an electrode array and find some way of digitizing the visual world and conveying it to the brain. But if you can convert the retina to a camera directly, that might be able to sort of short-circuit some of these aspects. And so here's a demonstration from Alan's group, and all the co-authors are down here. This is still a manuscript in preparation. So this is a mouse being put in a water maze. There's a bit of water in there to motivate it to move. And this is a blind mouse, and you can see it's not doing very well at navigating. It's supposed to go to this platform underneath the light. And it goes down an alley that's incorrect. And in the interest of time, I won't show you the whole video. But if we do one administration with a virus of the gene for this light-sensitive protein, then you see a very different picture. And you put the mouse in, and then you can see it now is doing much better. It can avoid obstacles and go to the platform. So the idea here is actually to try and see if this can be a clinical use to treat some forms of blindness. Ben Mateo, the acting CEO of this company, and myself have co-founded a company, EOS Neuroscience, to seek new treatments for blindness using this, and just received a first NIH SBIR grant. Back at MIT, we've been trying to figure out, is this molecule safe? We'll talk about viruses in a second, but the molecule comes from algae, and I'll show you some in a second from bacteria. Are these okay to put in the brain? So in collaboration with several groups, Bob Desmond's group and Grabel's group, and also pathologist Roderick Bronson, and we got advice from self-immunologists, we started to look at tissue that had been treated with these opsins, and also to look for antibodies in the serum, collaborating with a non-human primate physiologist, in order to see if there are reactions against these molecules. And so far, we haven't seen any pathology. Indeed, using viruses that are used in people, adeno-associated viruses, which most of us in this room already have, there are no symptoms, and it's already been used in 600 people in 48 different clinical trials without a single serious adverse event, which probably makes it safer than some drugs out in the market right now. You can start to do enduring multi-year labeling of neural populations with these optical sensitizers. So in some of our long-term studies, you're able to illuminate neurons over a period of time and not seen any degradation of the signals. These are work that we've done, once again, in collaboration with Bob Desmond and Grabel's labs, led by Shuang, to activate neurons with light and to see in the non-human primate brain over a period of many months, does the signal run down? And we see high-fidelity spikes throughout. Now, a second technology that we've been working on very heavily is that of optical neural silencing. And you might imagine that it'd be nice, both from a scientific standpoint, to be able to delete neurons for milliseconds at a time and to see what is lacking from the rest of the neural circuit. But also, there's many pathologies where we wish that we had the ability to silence overactive cells, such as in chronic pain and epilepsy and so on. So a few years ago, we found that there are actually light-activated chloride pumps that can be expressed in neurons, and they're safely expressed, and you can get hyperpolarizing currents. This is work that Shuang and I did. And here you can see some spiking, you shine light, and the spikes are deleted. You only get the stimulation artifacts. But the amplitude, as you can see, is not very powerful. The amplitude of the hyperpolarization is a lot less than the amplitude of the spikes. So we started a quest for exploring ecological and genomic diversity in order to see if we can find additional molecules. And we started to do screening for molecules from all over the world, from Western Australia, from Argentina, from southern Spain, that could yield higher currents, and we found some. So this is an example of a sort of a digital off-switch now, where we can shut down about 99.5% of the spikes that this neuron's firing while we turn the light on. And the neurons look great. They're not suffering from the expression of this molecule, although it's from an archaebacterium. We've also started to discover that you can both actively shift the spectrum of these molecules and discover molecules that are sensitive to different colors of light. So here are two populations of cells, one of which is silenceable with red light but not by blue, and one which we can shut down with blue but not by red. And so the idea is we can start to delete projections out of a single brain region to two different targets, let's say from a prefrontal area that can guide movement as well as regulating emotion and so on, and to independently perturb them millisecond by millisecond to understand when they are needed. Now, from a hardware standpoint, one of the problems with light is that, especially visible light, which is what I showed you so far, is that it doesn't necessarily go very deep into tissue. And so Jake Bernstein in the lab decided to try and figure out if there were a way of developing very high-density arrays of optical fibers where we could implant them and use them to systematically turn on and off parts of the brain. You might think of this as enabling what might be some of the first high-throughput screening technologies for the intact brain, where you can go through and quickly delete each one in turn and find out when they are needed. So this is an animation Jake made of what this might look like, with arrays of optical fibers implanted into the part of the mouse brain involved with learning and memory, and also involved with epilepsy. And recently we've been able to actually fabricate these things. So this whole thing is only about 8 or 9 millimeters wide, and can beam light under independent control to a dozen or 2 dozen sites in the brain. And we're now working on getting this up to even higher levels of maybe even hundreds or thousands of points of control. Now, in combination with another technology, this allows you to do some pretty interesting things. So Stephanie Chan, who was an undergraduate in the lab, figured out how to build fluidic injector arrays so we could deliver the viruses for these genes to multiple connected brain circuits because, as I mentioned in the first slide of this talk, the brain is three-dimensional and complicated, and we have to be able to sensitize precise circuits that connect points in the brain to light in a way that is meaningful from a behavioral, neuroscientific, neurological, or psychiatric standpoint. So on the left here, you can see one of her triple injector arrays that we made just for the sake of testing. This is a paper that's going to come out soon. And you can see here 3 different points in the motor cortex, which we've labeled so that they can be independently controlled with those fiber arrays that I showed you on the previous slide. Now, invasive implants are already used quite commonly in the electrical form for neurology and psychiatry. It's been estimated that well over 100,000 people have implanted electrode arrays for cochlear stimulation in order to provide auditory information to people who've lost their hair cells. 30,000 people or more with Parkinson's disease, deep brain stimulators, and maybe tens of thousands of others who have spinal cord stimulators and other stimulators. I've even seen a neurosurgeon who's doing Tourette's syndrome implants in adolescents. So as people start to test this, and we're getting into the hundreds of thousands of people, it's interesting to think about whether we can take this in novel directions using our optical and other strategies. So here's an example of work that Jake Bernstein and Mike Barada and colleagues are doing in the lab, along with Emily Coe, who wrote the software to program these arrays, and others. And this is all also in collaboration with Kai and Goosen's lab at the McGovern Institute at MIT. And what we did was label these viruses, different prefrontal cortical regions, with light sensitization so we can activate them with light. And that's interesting because these cortical areas can, in principle, project to subcortical areas, such as the amygdala, thought to be involved with many fear, aversive, and anxiety-related behaviors, and to control them through neuromodulatory means. So we implant the fiber arrays to hit these regions. And here's an example of a result where we do classical Pavlovian fear conditioning. The animal's in a box and learns to associate a tone with a shock, one of the most classical neuroscience paradigms of the last 100 years. And used by many as a sort of rudimentary, obviously imperfect, but we're starting somewhere, model of post-traumatic stress disorder, where a neutral cue becomes associated with an aversive state. And the optically modulated cortical mice are getting better, while the controls, even though they're being exposed to a neutral stimulus, this is called exposure therapy. You give the animal the same neutral stimulus over and over again and hope that it becomes neutral again, is not getting better. And so it's an example of the kind of thing that we hope to do at the interface between cognition, emotion, movement, and sensation. We've also recently started trying to work on non-invasive means as well. So transcranial magnetic stimulation, or TMS, has been around for almost 25 years, and just last fall, in 2008, it was approved by the FDA for treating drug-resistant depression by stimulating the left dorsolateral prefrontal cortex, this area here. So we've also started thinking about, can we start to improve the targetability of this technology by, for example, sculpting fields to make it more precise and to be able to target deeper structures and in different patterns. Now, I led off by the idea that we want to start developing brain coprocessors, that is, synergistic devices that can read out and deliver information. And what I've shown you here is just really the first baby steps in that direction by developing hardware that confronts the three-dimensional complexity of the brain and can drive information in very precisely. Nevertheless, we have started thinking about how do we generate hypotheses about the brain. And in collaboration with many other groups, can we start to build intelligence so they're not just passively or, let's say, in an open loop fashion perturbing activity, but building ways of augmenting these cognitive and emotional circuits with real intelligence. To that end, we've also started trying to develop ways of teaching students to think about these problems. So I started teaching a class called Principles of Neuroengineering a couple years ago, which students will make projects to try and develop ways to tackle these problems, followed by a lab class, Applications of Neuroengineering, and then finally, in collaboration with Jos Sponson and Rutledge Spenke, a class called Neurotechnology Ventures, which attempts to get people to think about how do you take innovations out of the lab and into the world in a difficult, long-term, high-risk, high-drama kind of space like neuroengineering. So I think I've acknowledged pretty much everybody as I described their work, but I wanted just to throw this up, and I think we have a few minutes for questions, I guess.
Very nice, thank you. Just curious, have you ever tried a control group where you put fiber optic photons when they don't have the rhodopsin?
Very good question. Yeah, the light that we're using is somewhat bright, and that's important because we don't want room light to be stimulating the brain. We do indeed do that. For our experiments, we haven't seen any compounds yet, but if somebody were to be using, for example, a visual behavior or looking at, trying to delete brain regions to understand how visual perception happens, the light might be a potential confound. We think we've been able to reduce that by coating the device as much as possible, but some light might leak through the brain back up towards the eye just internally, actually.
Have you ever put the fiber optic into the ventricles?
We have not on purpose. It's a fascinating idea. It's interesting for two reasons to think about that. One is that the ventricles, the brain is very reflective. The mean-free path of a photon is actually several millimeters before it gets absorbed, although it scatters very often. So the ventricles might actually be able to act as a waveguide, a very crude one, of course. But the second thing is also that the neurosurgeons have been using the ventricles as sort of a way of making neurosurgery cheaper and more efficient. So the Amaya reservoir used in cancer therapy is a very tantalizing way that you could cut the cost of neurosurgery down by a factor of several hundred potentially. So we've thought about that, but we haven't yet taken action upon that front.
Hi, Ed.
Yes.
I have a bit of a technical question. The method you're using is really great for activating specific types of neurons in particular regions of the brain, but if you wanted to be really specific, if you wanted to go to a very high resolution of which neurons you wanted to activate and which you didn't want to, how precise could you be with this method?
So two groups have actually used the very same light sensitizers with two-photon microscopy and have published papers where they can steer a beam and try to hit the boundary of a cell. One from UC Irvine and one from Princeton, Dave Tank's group. In both cases, I think there's a lot of promise that we can use infrared photons, two-photon microscopy, which is not time to describe here, to hit deep targets. And indeed, they've shown that, Dave Tank's group has shown that the two-photon cross-section is actually quite high. In principle, that would allow the addressing of stimulation at the single cell level within, say, a cubic millimeter or two per beam. Sure.
Yeah, my name's Rick Shuang. I was interested in your teaser in the front about improving the brain, and the list of characteristics to be improved do not include such things as kindness, ethics, justice. Now, I'm of the contention that we have enough dangerous features in us: territorialism, groupism, violence, aggression, that if we go down the self-improvement route with us to the point of going into the singularity with that, we are in a world of trouble. Okay, the first people to achieve takeoff will be the new tyrants.
Mm-hmm. You want me to comment on that?
Yes, I do. Are you willing to consider putting more of a focus on improving our character, the features that are so dangerous, than our speed and power?
Well, we try and think about the brain as an integrated system, and all these things are linked. So far, we're doing very early preclinical work on animals where we can't ask them what they're thinking. And that's just the way biomedical research goes. But definitely as we go forward with this, it's very important that we keep attuned to these things. For example, the very same circuits that we're working with between cortex and amygdala, for example, are also implicated in trust and in fear and in trustworthiness as well. And so it's quite possible that these technologies will at least point to the way of thinking about those problems at a deeper level.
What is the scalability of your research, and how long do you think it will take to get to a point of tens of thousands of neurons, or hundreds of thousands, or millions?
So we have a collaboration with Cliff Fonstad's group. His name is up here, but I didn't show any data from the collaboration yet. With a grad student, Anthony Zorzos, who's spearheading this, where we are trying to build massive arrays of waveguides, and they're not quite there yet. We have a presentation at the neuroscience meeting in Chicago in mid-October on it. But those in principle get us to the thousands range. At that point, it becomes really something of an interconnect problem, even just to control that many things. The devices should be fairly simple to make. But wiring things up will be hard, I think, for the foreseeable future. And we're trying to actively work on that.
That's all I got time for.
Okay, I'm being told to go away, so thank you so much.